Good afternoon, everyone. Can you hear me? Yeah. I know it's after lunch, and I myself am feeling a bit sleepy after that quite large portion. But I hope to take you through some of the issues we've faced developing some of the studies we do in the developing world, in particularly sub-Saharan Africa. The title of my talk is Closing the Data Gap in Low Resource Settings. Now, that's quite a broad topic, and that's not exactly what I'm going to be doing here today. What I will do, I'll try and highlight some of the issues around the data gaps that are currently out there, and use an example of one of the studies that we have conducted looking at drug-drug interactions with antimalarials and HIV as an example. So before we actually get into the meat of the matter, if we take a step back, as a pharmaceutical company, most drugs are approved by stringent health authorities, so in particular the FDA and the EMA. And this is based on a data set of safety information that's been compiled over the product development, as well as through pivotal phase 3 studies. Now, most of these studies do not allow comorbidities, and if comorbidities are allowed, the patients are usually stable. So it doesn't necessarily represent the real-world setting. More importantly, I think, especially what we're talking about today around neglected diseases, very little data is generated in the resource-limited setting. Most studies are conducted, as you know, in the US or Europe, and developing countries don't usually feature high on that list. On top of that, there's a consequent to that, there's a paucity of information around co-infections which are endemic to those countries. Now, when do we start becoming aware of these issues? In particularly from post-marketing surveillance, once the product is introduced into the health system, this generates further additional safety data that we may not have picked up in the development program for the product. And then what also might become apparent is unknown drug-drug interactions that start occurring that were not expected. So if we use the context of HIV and malaria, you can see here from the slide, and epidemiologically speaking, HIV and malaria overlap. So there were roughly 35 million people living with HIV in the world, most of it in sub-Saharan Africa and tropical regions, and similarly so for malaria. What do we know about the co-infection between the two? We do know that in HIV-infected patients, there's an increased parasitemia with malaria, and this is usually due to reduced host response, which is associated with lower CD4 cell counts of usually less than 200 or 300, and this usually results in anti-malaria failure rates that are higher than in the HIV-negative population. In particular, pregnant women are at particular risk, so they have more symptomatic or severe morbidity from malarial infections. They get more significant anemia during pregnancy. There's placental malarial infection as well, which also leads to low birth weight infants. And then we try and add antiretrovirals and drugs into the mix. What then happens? So basically, you can see here from the slide that you get more adverse events, more morbidity. In particular, in HIV-infected patients, cotrimoxazole or Septrin is used prophylactically to prevent PCP pneumonia. And if you give a patient who has malaria sulfadoxine or with pyrimethamine, they also increase risk of sulfonamide-induced drug reactions. If we also look at HIV-infected patients on AZT and efavirenz, which are widely used in the context of HIV in first-line treatment, again you see increased morbidity through neutropenia and hepatotoxicity. So we're not talking mild adverse events here, we're talking quite severe toxicities. And then from an HIV treatment perspective, drugs like efavirenz and Kaletra and boosted atazanavir, which are all WHO-recommended first and second-line treatment options, it also reduces the levels of the antimalarial drug atovaquone with proguanil, resulting in higher treatment failure rates. So with this in mind, we thought, what about, as Janssen, we had, at the time we were conducting the study, 2 antiretrovirals in development. What does this mean for the possible interactions with antimalarial drugs and or new compounds then? As you're probably very well aware, Coartem is used as a fixed-dose combination to treat malaria for falciparum, Plasmodium falciparum, and it constitutes with artemether and lumefantrine. They come in tablet strengths of 20 to 120 milligrams, and the dosing treatment is relatively simple. 6 doses, initial dose, and then a second dose 8 hours later, and then twice daily dosing thereafter. Why are they given together? But basically, they have a very synergistic effect with artemether and its DHA, the active metabolite, affecting more parasitic clearance, whereas lumefantrine is associated with a better cure, but not necessarily parasitic clearance. So what are the potentials for drug-drug interactions? Artemether and lumefantrine are both metabolized by the CYP3A4 isoenzyme system in the liver, and DHA is further glucuronidated there as well. And then if we look at the newer antiretrovirals like darunavir and etravirine, darunavir being a boosted protease inhibitor and etravirine being a second-generation NNRTI, they're also using the same pathway for metabolism. So the interaction or the implication for further drug interactions is theoretically already there. Historically, what we do know, lopinavir, which is the WHO recommended third agent for second-line treatment, has been shown to decrease coartem, as well as increase lumefantrine, which is associated with QT prolongation and obviously some degree of cardiotoxicity. For nevirapine, it also reduces artemether and DHA and also increases lumefantrine. So again, issues with antimalarial potency as well as increased risk of toxicity with QT prolongation. So this is the study design. I'm not going to go through much detail around it, but basically patients were randomized into one or two panels. These are healthy volunteers. The study was conducted in Poland, and you might say, why would you conduct the study in Poland? Because antimalarials aren't probably going to be used in many Polish people. The intention was to do the study in Kenya, but because of feasibility testing, the site in Kenya was eventually not selected due to not complying with GCP standards. So we had to make a quick decision where else to do the study, where we had some experience before. So basically, patients receive coartem for 3 days, and you can see the dosing intervals there, and PK sampling was taken accordingly. There was a washout period for at least 4 weeks, and the healthy volunteers either receive etravirine or darunavir twice daily. And then at day 8, the coartem was introduced, and the PK sampling was taken along that time as well. So what did we find? Again, I said all patients were white, male, non-smoking. 5 subjects discontinued, 3 for adverse events, 2 withdrew consent. Most of the adverse events, luckily, were grade 1 or 2. And also, quite fortunately, the QT prolongation was not really seen in the study. So we looked quite intensively for QT prolongation because of the increased possibility of lumefantrine. You can see the 3 reasons for discontinuation being tendon rupture, gastroenteritis, and rash, none of which were thought to be due to study drug. So these are the results of the study from a PK perspective, and you can see in the first bullet that roughly 52% reduction in the artemer component of coartem by co-administering it with etravirine. That sounds like quite a significant interaction. What does that mean clinically? We don't necessarily know. But it is obviously something that needs to be borne in mind when prescribing etravirine with coartem. Similarly, if we go down the list for darunavir, it does increase lumefantrine levels by close to 3-fold, and that's probably being driven by the CYP3A4 inhibition. What does that mean clinically as well? We did look for QT prolongation, but we did not see any in the study. But the implication there is if you're giving other drugs in conjunction with darunavir and lumefantrine that can increase QT prolongation, you're probably pushing patients into the no longer therapeutic range of the products collectively. Interestingly, the coartem had no effect on the antiretrovirals, which is good. These slides just show the drug interactions from a PK curve perspective, and here you can see the bioavailability is decreased if you were to co-administer etravirine with coartem, and the artemether part of that is being reduced by the 50% I mentioned before. And similarly here, you see the increase in the PK curves above where lumefantrine levels are increasing by almost 3-fold. I just want to put a bit of a plug here. This is for the University of Liverpool. It's one of the websites we support as J&J. It's the HIV drug interaction website. They also have a similar one for hepatitis C infections. And it's very easy, user-friendly system where you can plug in what drugs you want to combine together, and it gives you the interaction or the possible interactions to expect and what you need to do when managing the patients from a treatment perspective. And here you can see with atravine, you get an amber signal showing a potential interaction which requires close monitoring and alteration of drug or dose timing of administration. So, in conclusion, atravine and darunavir both can be co-administered. Atravine in particular must be co-administered with caution because of the reduced levels of artemether, which may result in lower antimalarial effectivity. For darunavir, they can be administered as well without dose adjustment, but care must be given if prescribing other drugs that can increase QT prolongation at the same time. What are the limitations of the study? And I alluded to this earlier. The study was conducted in Caucasian males. There's no data generated in women, which is obviously very important, and especially what we saw at the beginning, where pregnant women can have severe anemia, severe morbidity associated with malaria and HIV co-infection. And then again, there's no data generated in children. And then in the real world, we don't give these drugs in silo with just one with the other. We give them in combination therapy. What does that mean? Those drug interaction studies have not been done, and so the clinical consequence of this is not completely understood. I'll stop there. Oh, and I'd like to acknowledge the participants in the trial.